last science seminar in that desert. We've had a lot of good science seminars this semester. That's been fun. Hope you guys have enjoyed your time this semester as well. Okay. Today, for our final speaker, we say the best for last. We have David Barondeau.
That's good.
Or Barondeau.
Barondeau. It's good.
Yeah,
that's good.
David Barondeau from chemistry at Texas A&M University. He's a biochemist.
Nice.
And we're talking about the difference between biochemistry and what was it, chemical biology.
Uh-huh.
And so maybe you guys can have and determine that a little later here today. So David grew up over in Utah. He got his bachelor's degree at the Southern Utah State College, and he went from there to Texas A&M and got his PhD. And from there, he did some postdoc work at the Scripps Institute, the Scripps Research Institute, which is a famous place for biochemistry type of people. And he was supported by some prestigious fellowships there. And then from there, he went to Texas A&M, and he's been there for a while. And he's now an associate professor, so I assume he's got tenure.
I do.
Yeah, so he's a tenured professor over there at Texas A&M, and he's published many things on this biochemistry stuff related to X-ray crystallography structures, biological things, and stuff like that. So we're really happy that David is with us today. After the seminar, we'll be having lunch over in the Joyce Room. So if you're interested in learning more about A&M, about biochemistry, all this kind of stuff like that, please join us for lunch. Let's give David a warm welcome.
Thank you for inviting me to come here. I've never been here before. This is actually quite nice. It was a nice little drive this morning. Almost no traffic. It was really beautiful. So, yeah. It's not that bad. I do get up early anyway, so I didn't really get up any early. So. And so this is the building I work in. This is the ILSB building. It's an interdisciplinary life science building at A&M. So we actually have, this building is actually separate from the chemistry building. And so the structural biology type of research that I do is actually housed in this building, because this is where the X-ray generator is and things like that. And so this is where I work. And so just hit the keyboard?
Yeah, try this keyboard over here. Oh no, what the heck? Things are funky sometimes. Let's click on here.
Oh,
there you go. All right, is it working now for you?
Yeah, now,
All right.
now it's working. Okay, great. And so I'm part of the graduate faculty at Texas A&M, and so we're always recruiting students interested in chemistry or biochemistry. And so if this is something you're kind of interested in, I suggest you look at us. We're just down the road. Stipends are about, are $27,000 for 12 months. We pay the tuition and fees for the students. And we guaranteed funding for 5 years of graduate study. The deadline is December 15th every year, so this year and every year. So whenever you were ready to think about graduate schools, this is when our deadline is. One of the things that we really look for in our potential students is research experience. So if you have the opportunity to do research with faculty here or do an REU, I would definitely encourage you to do that if you're interested in graduate school. Okay, so now I'm going to tell you a little bit about what we do, my group. We study iron sulfur clusters. And so these are protein cofactors. And so they exist in most life forms. There's different types of iron sulfur clusters. Here are the two most common types. This is what we would call a 2-iron 2-sulfur cluster. It's got 2 irons. These could be iron 3 or iron 2, and then 2 sulfides, S2-. And then these 2-iron 2-sulfur clusters are ligated to the protein through a bunch of ligands, typically cysteine residues, like is shown here. You could also make 4 iron 4 sulfur clusters. They look like a little cube. And these little iron sulfur clusters and these iron sulfur clusters then can do a variety of things that I'll tell you about in just a minute. There's additional forms as well. These can be part of more complex active sites. And they, one of the things that's characteristic of these types of clusters is they're oxygen sensitive. So in my research group, we do everything in anaerobic glove boxes. There's plenty of water in those anaerobic glove boxes, unlike some of my inorganic colleagues. But it's all done at 1 part per million or lower of oxygen. And so iron sulfur clusters, probably their most famous function is electron transfer. And so this is a picture of the respiratory complexes in your mitochondria. This is how you generate energy. And so these are the iron sulfur clusters, and they're involved in just transferring electrons through these complexes and generating a proton gradient, which you eventually use to make ATP. And so that's probably the most common function of iron sulfur clusters is just electron transfer. They're electron transfer conduits. However, there's a lot of other functions as well. They're involved in DNA repair, gene regulation, all types of primary metabolism, secondary metabolism. They have functions in substrate binding and activation, generations of radicals, and so forth. They're also involved in the synthesis of other cofactors. Okay? And so they're important for a lot of different things in the cell. And so what we're interested in my group is how you make and then distribute these clusters to all the proteins that need them to do these functions. And so how do you make an iron sulfur cluster? It turns out these things will spontaneously form. Okay? And so this is something that Dick Holm from Harvard spent a lot of years characterizing. And if you get the right combination of iron, sulfide, and ligands, you can make all these different types of iron sulfur clusters that are found in biological systems. And they'll just self-assemble. Okay? If you have the right stoichiometry, they'll just self-assemble. And in fact, if you take a protein in the apo form, which means it doesn't have the cofactor, and add iron and sulfide, they will spontaneously build clusters in solution and insert them into the proteins and activate the proteins. Okay? And so this is all pretty spontaneous type chemistry. And so one question you might ask is, why do we study it then? And it turns out that there's biosynthetic processes that do the same chemistry, and that's what we look at. And so it turns out that the biosynthesis of iron sulfur clusters occurs in bacteria, it occurs in eukaryotes, and there's particular pathways to build these clusters. So the reason we need to build them is not, they will just spontaneously form, like Dick Holm showed, but the reason we don't do that is it turns out that the substrates to build those clusters are actually toxic. Okay? And so iron, too, is prone to fit in chemistry, making hydroxyl radicals, which damages DNA and lipids. And then sulfide is actually a gas, and it's an inhibitor of cytochrome oxidase. And so instead of just making sulfide and iron and letting them self-assemble, we have a pathway that builds these clusters specifically and then delivers them to the targets. And so two of the big people in my field for characterizing different proteins and pathways are Dennis Dean, he's kind of the godfather of this whole field, and then Roland Lill, he's kind of taken over and done a lot in the eukaryotic systems. Okay. And so it turns out that these different pathways have a lot of similarities, and I'm going to show you a little bit about the mitochondrial version. Okay, so this is a very simplified diagram. And so when you want to build these types, this represents an iron sulfur cluster, a 2 iron 2 sulfur cluster. And when you want to build these, what you have to do is you have to provide sulfur, you have to provide iron, you have to provide electrons. The sulfur actually comes from the amino acid cysteine. And this enzyme is a PLP, a pyridoxal phosphate containing enzyme. I'll talk a little bit more about that in a few minutes. And this will convert cysteine into alanine, essentially pluck the sulfur off. You'll make a per-sulfide intermediate, so a sulfur-sulfur bond. And then this sulfur will be transferred to this scaffold protein. These proteins actually make a complex. And then once it's transferred, you can incorporate iron and electrons and build these clusters. And so that's kind of this simple version. And so this cysteine desulfurase, in eukaryotes consists of 3 proteins. The names aren't so important here, but it turns out that this NFS1 is the catalytic subunit. The ISD11 and the ACP are additional accessory proteins that, interestingly, don't function in the cysteine desulfurase for all the bacterial systems, but do in the eukaryotes. And so when I first started this project, that was one of the big questions, is why does the eukaryotic system have this ISD11 protein where the prokaryotic system does not? And another big question was, this FXN is also known as frataxin. This is required for iron sulfur cluster, or stimulates iron sulfur clusters in eukaryotes, and doesn't in bacteria. And so there's these different additional proteins that are involved in the eukaryotic system that aren't involved in the bacterial system. And so one of the questions that we had when we started was, what are the roles of these proteins, and what are they doing? Okay. And so this ISD11 protein is a member of what we call the LYR proteins. And so this is a newly discovered superfamily. It's about 12 eukaryotic proteins. They're all in the mitochondria. This is a cartoon of the mitochondria. And they're involved actually in all kinds of respiratory type events. So these are the respiratory complexes I showed you earlier in like the second slide. And these LYR proteins are actually subunits or assembly factors to make these function. And this is the complex that I'm going to study, or we study down here, and it also has one of these LYR proteins. And then it turns out that acyl carrier protein binds to all of these LYR proteins in the cell. And so that was a huge shock. Okay, so what is acyl carrier protein? So it turns out that acyl carrier protein is actually a well-known protein. It's found in fatty acid biosynthetic pathways, and it's also found in some natural product biosynthetic pathways. And so acyl carrier protein is a neat protein that has this phosphopentathionein cofactor, and then you build fatty acids on the end of this. And so normally this is involved in building fatty acids. So what is it doing in iron sulfur cluster biosynthesis? That was a huge question. We had no idea when we started this. And so I'm going to show you a structure in a little bit where we now have a clue of what this is actually doing in this pathway. It turns out that in the mitochondria, you have this acyl carrier protein. You also have another acyl carrier protein in the cytosol that's part of the fatty acid biosynthetic complex. And so this mitochondrial acyl carrier protein really isn't involved in making so many of the lipids for the membranes. It's actually involved in making lipoic acid, another cofactor, and actually this function of interacting with these LYR proteins, which I'll tell you about in a little bit. Importantly, if you knock this protein down in the mitochondria, what happens is you lose iron sulfur cluster assembly. And so this was discovered about 3 years ago, I guess now. And this was a shock to the iron sulfur community because we didn't really expect this to be involved. Okay, so here's another picture of a mitochondria. And so one of the things that is important for funding in this area is it turns out that almost every one of the proteins involved in iron sulfur cluster biosynthesis and then delivery, and then this is a cytosolic iron sulfur assembly system. A lot of these different systems, or a lot of these different proteins, defects are directly linked to disease. Okay? And so that helps when you're trying to get NIH funding or funding from other health-related agencies. I'm going to tell you specifically about this frataxin protein here today. Okay, so what is frataxin and what disease is it related to? So frataxin is actually related to this disease called Friedreich's ataxia. This is incurable, it's lethal, it's a neurodegenerative disease. Symptoms typically begin between the ages of 5 to 15. And typically for these people, it looks kind of like they're drunk because they're stumbling and they have ataxia essentially. And that's kind of the first thing you see. Eventually, this progresses, and they often die from heart disease, actually. And so, the way that works the way the disease actually works is in the first intron of the frataxin gene, there's this GAA nucleotide sequence, and this gets repeated over and over again. It's a nucleotide expansion disease. There's quite a few of these. This is one where this GAA is repeated, and people with Friedreich's ataxia have between 70 and 1,700 of these repeats in their first intron. And what this ends up doing is lowering the frataxin protein level in the cell, essentially. Okay? And the longer the repeat, the lower amount of frataxin protein you have, and the worse the disease and the faster it progresses. And so when you have low levels of frataxin protein, what happens is the iron sulfur clusters in the cell are all depleted. And so that was the initial connection between frataxin and iron sulfur cluster biosynthesis, is this. And then once they're depleted, another thing happens. You get actually iron accumulation in the mitochondria. Apparently there's some signal that's iron sulfur base that says quit pumping iron into the mitochondria, and that's lost when you don't have frataxin. And so you keep pumping iron into the mitochondria, and you make plaques, and then you actually start to get oxidative stress through Fenton type chemistry. Okay? And then once you get oxidative stress and reactive oxygen species, this degrades the iron sulfur clusters, and so this becomes kind of a vicious cycle. And so what ends up happening is these patients then have defects in their ability to do all the things that iron sulfur clusters participate in, generating ATP, DNA repair, all those things are compromised. And so what pharmaceutical companies have done is, we knew this all the time, is that once you lose these iron sulfur clusters, you get this iron accumulation and oxidative stress. So they've tried chelators and antioxidants as therapies, and those have all failed, actually. And they've probably failed because you're treating one of the symptoms rather than the root cause of the disease. And so what our job as biochemists is to figure out what frataxin actually does in this step. Okay? And so I'm going to tell you a lot about that for the rest of the time. Okay, so first of all, how do you monitor iron sulfur clusters? You could do this with a variety of different techniques. The most common are just absorbance. These are brown colored materials due to the sulfur to iron charge transfer bands. And so you can monitor the increase in absorbance. This isn't so nice because a 2 iron 2 sulfur cluster bound to one protein and bound to another protein, their absorbance spectra look almost the same. And so you can't really, if you want to look at transfer events, you can't really tell if it's transferred. CD spectroscopy is really nice, visible CD spectroscopy. So this is circular dichroism. So you're looking at left versus right polarized light. And so you're looking at the same charge transfer bands, but now if the environment around the cluster is polar, then you'll get a CD signal. Right? And so these 2 iron 2 sulfur clusters, the protein environment around these clusters is chiral, and so I'm thinking polar chiral, and so you get this nice signal. Okay, and so you can develop a 2 iron 2 sulfur cluster signal over time. Okay? And so you can monitor these 2 iron 2 sulfur cluster synthesis events using CD spectroscopy. Okay, and so for human frataxin, most people would agree that it's involved in iron sulfur cluster biosynthesis. If you lose the frataxin, you have decreased iron sulfur cluster activity. And so the first thing that we did is we generated an assay to say, well, if you add frataxin to this assay, does it increase the rate of iron sulfur cluster synthesis? And so we monitored this with ellipticity, so CD spectroscopy. And if you just look at the red and the blue, ignore the other two for now. What you'll see is this one is in the absence of frataxin. When you add frataxin, you increase the rate pretty dramatically. One other thing about the Friedreich's ataxia is the frataxin levels are really sensitive. So if you have 40% of the normal levels of frataxin, you're okay. If you drop below that, there's some threshold. Once you drop below that, you're not okay. Okay? And so it's not a real dramatic difference in the amount of clusters, but there is some threshold level. Okay, and so it definitely seems that you can stimulate this iron sulfur cluster assembly by adding frataxin. So then the next question is, well, what step does it actually affect? And so this is back to that original diagram, and we had it written here as frataxin. And so what we wanted to study then is ask the question, does frataxin actually accelerate the chemistry of this enzyme? Okay? Because we didn't really know. All we knew is that if you added cysteine, electrons, and iron, you'd be able to iron sulfur clusters a little faster. But it could be that this actually accelerates this enzyme. And actually, it turns out it does. Okay? And so that was kind of a surprise as well. And so if you look at the cysteine desulfurase and how that works, this has a pyridoxal phosphate cofactor, and that's what's actually drawn down here. This red part is the pyridoxal phosphate. And the cysteine substrate binds through its amino terminus to the PLP, and this acts essentially as an electron sink for the chemistry that goes on. Okay? And so at one step of the reaction, a cysteine on a mobile loop, it's actually represented by this sulfur here, comes down and attacks the, well, this ends up being a ketamine intermediate and making a persulfide, and then eventually this will become alanine a little bit later in the pathway. Okay, and then you generate this persulfide, and then you can cleave this and use that sulfide to generate methylene blue and measure activity, and that's what we do. You can also, once this is generated, this sulfur could be transferred to the scaffold protein for cluster synthesis. Okay? And so one of the assays we did is we then added frataxin and asked the question, does cleavage of this bond and generation of methylene blue occur faster in the presence of frataxin? Okay. And it turns out it does. Okay. So this ISCS is actually the enzyme from bacteria. Okay. And so this is kind of the, this is a fully functional enzyme that doesn't require any of these additional proteins. Doesn't require this ISD11, it doesn't require frataxin. And so this is the activity of that enzyme. So this is the doing steady state kinetics. This is the KCAT and KM. It's a modest enzyme. It's not a great enzyme. If you look at the human enzyme, this NFS1 homolog, it turns out the NFS1 by itself has no activity at all. If you add in this ISD11, you have very poor activity, actually. Very high KM for cysteine and a very low KCAT. It turns out that this activity is low until you add in the frataxin, and then you stimulate this low KCAT by an order of magnitude. Okay? And so that activates the system. Okay? It also turns out that the KM is also affected during that step. Okay? And so what we showed with this study is once you add frataxin, it binds to the cysteine desulfurase complex and accelerates the chemistry, the cysteine turnover chemistry, and then subsequently accelerates the iron sulfur assembly. Okay, and so at that point we had a couple of questions. Okay, first question is, why does the cysteine desulfurase activity for this complex so low? It's very low. Our nomenclature is horrible. I'm not going to make you. This is the nomenclature in the literature, but this is the cysteine desulfurase, this SDA complex. This also has acyl carrier protein. We didn't know it at the time, but it did. And so this has very low activity compared to the cysteine desulfurase of the bacterial system. And so one of the questions is, why is that activity so low? And these catalytic subunits have 60% sequence identity, which is actually quite high. Okay? And I'm going to show you a little bit later the structure, and the structures of those subunits look almost identical. Okay, so the question really is, why does it have low activity, and then why does frataxin stimulate that activity? And so we did some additional experiments. A student in my group, Sachin Patra, who received his PhD last year, what he did is he did some radial labeling studies. And so what he did is he added radial labeled cysteine, and then we could monitor the sulfur incorporation into NFS1, this is cysteine desulfurase catalytic subunit, and then transfer to the scaffold protein, which is going to then build the clusters. And then we could ask the question, if you add frataxin, does the rate of sulfur incorporation in these two proteins increase? We quenched with acid, and then we analyzed by separating the proteins and then doing some quantitation. And so here's kind of the scheme of what we thought was going to happen. This was the NFS1 and the ISU. These are the cysteines on those proteins. You have the radiolabeled cysteine, this is the red. You could put a radiolabeled, make a persulfide on NFS1. This could transfer to ISC2, which would be this step here. And then you could actually incorporate a second sulfur through another round of catalysis. And so we fit our data to this scheme. And it turns out that here's the actual data. These are in the absence of frataxin, this is in the presence. And what you could just see qualitatively is when you add frataxin, the black lines are the rates of sulfur incorporation in the NFS1. You can see this rate is a little bit faster than that one, and that this rate is much faster than this one. If you actually fit the data to a kinetic model, it turns out to be about 6.5-fold faster for this step, transferring or building a persulfide on NFS1, and about 30-fold faster for transferring it to ISCU. Okay. So that suggests that it's somehow affecting the chemistry of the PLP, frataxin affecting that chemistry. So then we did another set of experiments. We looked at the mechanism in a little bit more detail. The only part I really want you to look at here is this is where you start. This is the PLP at the beginning. It has a particular absorbance maximum. When you add cysteine, you can make this gem diamine. This occurs pretty quickly. And then after that, you make a aldamine, quininoid, and ketamine intermediates. And these all have distinct absorbance maxima. And so you can monitor those individually if you had some rapid kinetic approach. And so that's what we did. We looked at how quickly do these intermediates form and decay. And so to do that, we use stop flow kinetics. And so essentially what you do in this is you put your proteins in one syringe, your substrates in the other, you mix, and then you get into this mixing chamber with a stopping syringe, and then you can measure things on the millisecond time regime. Okay? We did all this anaerobically in a glove box. And so one of the key results that we found is that one of the intermediates is greatly affected by frataxin. Okay? And so in red is the decay of this quininoid intermediate. And so it's pretty slow in the absence of frataxin. When you add in frataxin, it actually, the formation's the same, but the decay is much faster. So somehow frataxin is accelerating the chemistry from going from this quininoid to this ketamine step. Okay? And so how could it do that? Well, the chemistry that's going on is you're just protonating this intermediate. And so what it suggests is that somehow frataxin is positioning this proton donor for function. Turns out that it's been proposed that this proton donor is that same mobile loop cysteine that is involved in transferring the sulfur to the ISCU. And so what we think frataxin does is it comes down into the active site, or it binds, and then this mobile loop comes down to the active site. This mobile loop has a cysteine residue. It donates a proton. That donation of the proton advances the quininoid to the ketamine intermediate. Once you're at this ketamine step, now that mobile loop is now deprotonated and can then attack the terminal sulfur of the ketamine intermediate to make a persulfide. And so we showed that this was accelerated with our radiolabeling study. And then once you've made that, you can then deliver that to ISCU for iron sulfur cluster assembly. We also showed that was accelerated. So what it appears is that everything involving that mobile loop is accelerated by the addition of frataxin. And so to test that further, what we did is we mutated that mobile loop cysteine to an alanine. And if you do that, this intermediate doesn't decay at all now. It's just stuck. And so you no longer can donate the proton to advance the quininoid to the ketamine. Okay, so this is further evidence of that the mobile loop cysteine is actually the proton donor for that. so summary of this part, this mobile loop cysteine, this S transfer loop, functions in 3 ways. It functions as an acid to protonate the cis-quininoid intermediate, a nucleophile to cleave that bond, and then a carrier to deliver that sulfur to the acceptor protein. And then that mobile loop cysteine, each one of those functions actually is accelerated by frataxin. So somehow frataxin is affecting this trajectory of this mobile loop. So that's really the story here. So now what we wanted to do is, well, let's get some structural information for this. Okay, so we have some idea of how this might work. Let's get some structure. Okay, so if you want to get the structure of the human cysteine desulforase complex, what we do is we often do these at a synchrotron now. We have a home source at A&M where we collect some initial data to see, to screen conditions and get good diffracting conditions and cryo conditions. And then we have beam time at the synchrotron. And then what we do is we mount our small crystals on a loop, put them in a cassette, and mail them, frozen, to the synchrotron. They put them in this dewar, and then there's a robotic arm that will go into the dewar and take the loop with the crystal and mount it on the goniometer. And then we control that all from A&M. And so, and then we can collect the data. And so what you do in this process is you generate a crystal. So this is one of the hardest part of protein crystallography, is generating the crystal. And so once you've generated this crystal, then you hit it with X-rays. We don't really use an X-ray tube anymore. We use a synchrotron beam, which is much brighter. And then once that beam, X-ray beam hits the crystal, you're going to get some constructive interference, and you're going to get a diffraction pattern. And so this diffraction pattern, then, you will do this with different orientations of the crystal and collect a bunch of these diffraction patterns. And it's going to turn out that where these spots are on this detector will tell you how the crystal is, how these different molecules are packed into the crystal, the symmetry of the crystal. And then the intensity of these spots will tell you something about what's inside each one of these unit cells. And so then once you've generated a bunch of these diffraction images, each one of these will have a structure factor, this FHKL. You can then use this function to actually generate an electron density map. Okay? And so a little bit of difference between protein crystallography and small molecule crystallography is there's a lot of manual intervention at the protein crystallography level. For small molecules, you don't really do any fitting. At a protein crystallography, you do. So what you end up getting is something that looks kind of like this, like an electron density map. And then the job is to then fill in the model like it's been done on this side. Okay, and so you build in the side chains and the main backbone, and then analyze statistics to tell you if you fit the structure sufficiently. Okay, so we did that. So here's a crystal of our complex. Here's actually the crystal mounted on a loop in a diffraction pattern. And so this was actually done by Seth Corey in my group, who recently got his PhD as well. This was actually the first crystal structure of mitochondrial cysteine desulforase. And so when you look at the actual, you build the model and you refine it, and then you analyze the structure in the end. When you look at it, if you compare the human version, a monomer of the human version versus the bacterial version, in a cartoon-like image, these look pretty much the same. Okay? There's some minor differences, but not any big differences. This really doesn't explain, I mean, this was kind of what you'd expect for the 60% sequence identity, but it doesn't explain at all why this version is fully functional and this version requires the other 3 proteins to be fully functional. And so you don't start to get that information until you look at the overall complex. If you look at the overall complex, this is the structure of the human NFS1 ISD11 with ACP. And so this is the overall structure. And so you can see this is one of the catalytic subunits of the NFS1 that I just showed you. Here's another one. This ISD11, which is critical in eukaryotes but doesn't exist in prokaryotes or bacteria, This is actually mediating all the interactions between the two halves of this assembly. And so that was a real surprise, because cysteine desulfurases typically have a homodimeric nature where this subunit and this subunit would be on top of each other. Okay? And so this was really shocking, actually. And then the ACP I'll talk about here in just a little bit. We actually did a negative stain electron microscopy structure because this was so unexpected, actually. And so what we wanted to do was prove that this wasn't some artifact of crystal packing in the crystal, and that this also exists in solution. And so this is electron microscopy structure, so it's consistent with the crystal structure. If you look at the active site, so this is the NFS1 active site, and this is the bacterial version, the active site looks identical too. So it still doesn't explain why one is fully functional, one is not. And so if you start to look at it though, the bacterial version has these other parts of the structure. These are actually from the other subunit when it forms that homodimer. And so now it's starting to make a little bit more sense, right? So this form doesn't have those other interactions, and so you can kind of imagine why this form doesn't have, the mobile loop trajectory is a little bit messed up. Here you can imagine the mobile loop coming right down to the PLP to do the chemistry and then transferring over. And these interactions from the other subunit kind of guiding that trajectory down to the active site. Whereas this one doesn't have those type of interactions. Actually, I'm going to skip that. If you look a little bit closer at the ACP part, this is also really interesting. It also explains why we have ACP in this complex. And so this is the part of the ACP unit, and this is the phosphopentathione, along with the electron density. This little stuff here is the electron density. And then you can see that the acylated part is stuck down into the core of this 3-helix bundle. Okay? And so it turns out that the ISD11 is a 3-helix bundle, but it has an empty core, right? It doesn't have hydrophobic interactions in the core. That's really unusual, actually. Normally a protein will have hydrophobic residues in the core of the protein, and that will exclude water, and that's part of the protein folding mechanism. This one doesn't have that. It has an empty cavity. And so this, what you would expect is in the absence of this, that this protein would be highly unstable, and that's actually what you see in the cell. It precipitates. And so what the job of ACP is to stick this greasy lipid down into the core of this ISD11 and provide the hydrophobic core for this other protein. We'd never seen that kind of interaction before. Since then, all those other LYR ACP interactions, a lot of the electron microscopy structures that have come out have shown exactly the same type of interaction, where they stick lipids down the center of these 3-helix bundles as a mechanism to stabilize those LYR proteins. And so it also turns out that this is the LYR, or the ACP down here. This is the PLP up here. And there's actually hydrogen bonding interactions across this interface. And so it's easy to explain then why ACP affects the substrate binding up here through these interactions, through these hydrogen bonding interactions. And in fact, some of these residues are actually clinical mutants for NFS1 that lead to another disease. And so there's really an important network of interactions between the ACP and the PLP. Okay. And so overall, what does this ACP do? Well, this is another cartoon of the mitochondria. And in the mitochondria, you have acetyl-CoA. Fatty acid synthesis can then make octanoyl ACP. So this is an 8-chain carbon unit. And then this is an intermediate to make lipoic acid for the function of these enzymes. But you can also make longer chain ACP units. And these longer chain ACP units are the ones that actually interact specifically with all these LYR proteins. And so there's some unknown regulatory mechanism in the mitochondria that we're just now discovering, actually, that allows acetyl-CoA then to control respiration. Okay? And so I think this is going to be a really big story in the future, probably from more genetic type groups than our group, but this is going to be a big story. Okay, then it gets more complicated. I know. It always gets more complicated. Turns out that there's another structure of the complex as well. And so this is the structure that Seth and my group determined. Another structure was also determined with exactly the same mass. Same subunits, same composition. Actually, the same constructor is used to make both of them. Okay? So then you ask, well, what is going on, right? Is one of these an artifact? If you overlay the NFS1 ISD11 ACP units from both of them, that's what's done here, they overlay almost exactly. So the only difference is how these two units come together to make these two assemblies. So you can make these two different assemblies by coming together across two different interfaces. If you come together across an NFS1 interface, you make this form, which we call the closed form. If you come together across the ISD11, you make this form, which we call the open form. So then you immediately ask the question, well, what's the relevance of these two different forms, and how does this work? The first thing we did is we repeated, since this structure was actually done by another group, they had slightly different expression and purification conditions. We didn't think that would make a difference, but we decided to make the material exactly the way they described it, and then measure activity. And so that's what we did. And so this was our method, this was the other method, and basically the activities are the same. So making the material in the two different ways makes no difference. Okay? And you can also crystallize the complex in either form using either batch of material. So that was kind of interesting. And so that's essentially what this is. And so then Seth did a really nice experiment. This was a tough experiment. So he grew crystals of each of the different forms, the open and the closed form, and then he washed those crystals and then dissolved them and then measured their activity, with the idea that maybe you could get a pure open and a pure closed form, and they would have a difference in activity. You could tell which one was the active form and which was not. They have exactly the same activity profile. So then that means one of two things. It means either that these two forms are in equilibrium, which is our favorite, or that both forms are activated by frataxin in exactly the same way, which we find not so reasonable. And so it turns out that we think that these two forms are actually in equilibrium, and this is part of a regulatory mechanism, is what we think. And so we used one more approach. We used ion mobility mass spec. And so in this, what you do is you ionize the sample, you pass it through a drift tube. Depending on the conformation of the protein, if it's more open or more closed, it will go through this drift tube at a different rate. Okay? In the end, you'll get the same overall mass, the mass to charge, but if they have different shapes, they'll go through this drift tube at different rates. Okay? And so this is really a nice technique for looking at different conformational states of proteins. And it turns out that we could see two different states, and then when we add frataxin, it all switches to one state. That's essentially what I want to tell you. I'm going to skip this one too. And so what we think is going on is this is a morphine-like model. And so you have an equilibrium between two different states, and some molecule will bind and shift the equilibrium towards one or the other. And this is well known in the literature, okay, for a bunch of different enzymes. What's different from those systems and our system is these typically will have different oligomeric states, like a trimer and a tetramer. Whereas in our case, they're the same size. They're just different interfaces. So this is what we think is going on. You have an open form and a closed form. We think they could dissociate into NFS1 ISD11 ACP units, which can be in equilibrium. And we think frataxin binding will switch this equilibrium to the closed form. So that's kind of our model. Okay. And so this is Seth's picture. So this would be the open. Oops. Maybe. If this is going to work. Ah, it is. And so it's transitioning from that open form to the closed form to kind of get you a picture of this transition. And then we think once it gets to this closed form, frataxin binds and stabilizes this form. I guess it's not going to do that part. Okay. Yeah. frataxin then would bind and stabilize the closed form and activate it. So overall, what we think is happening is you have two different states that open in the closed form. The open form is the stable form, and it's the inactive form. And when you bind frataxin, it switches to this closed form and activates the complex. And that process is actually the frataxin activation system. And so what that means is, if you have this type of equilibrium, what you need to do if you want to make a drug is you need to find molecules that bind and stabilize this closed architecture and drive this equilibrium towards this form. Okay? And that's what you'd have to do. So typically when you do drug discovery type of things, what you're trying to do is inhibit enzymes. It's easy to kill things. In this case, you're actually wanting to activate things. And so it's harder to make a drug. Okay? Here what you'd have to do is make something that will bind and activate by shifting this equilibrium. Okay. So this is my group, both graduate students and undergraduates. And then these are some of the former students that were in my group and where they are now. This is my current group. This is the funding that I'm very grateful for. These are some of my collaborators on this project. And if you have any questions about my research or the research at Texas A&M or the graduate program, you can email me at this address. I'd be happy to answer any questions.
Questions for Dr. Barondeau, biologist here. Dr. Baller, biologist. How
One over
stable
there.
are these things to do X-ray crystallography on?
How stable are they?
Yeah,
Well,
do they denature real fast?
they do, and so are reasonably fast. And so we do a lot of our, we have to prepare all the material. We prepare the materials in an anaerobic glove box at lower temperatures, so we have a cooling system for that. And then we set up the crystals actually at room temperature. And so they last long enough to generate crystals, is what I would say. But they require a lot of reductant. We have a lot of TSEP as a reductant for the sample. I think it's a question over there.
I just have one question. You mentioned earlier that the DNA sequence is a repetition of a certain group of nucleotides, and that's what deactivated it. Is that like a feedback loop?
So that's like a replication error, a DNA replication error. Yeah, and so we all have a certain number of those GAA repeats. None of us just have one. And so what happens is if you get too many of them, your frataxin levels get too low. And even if you have one frataxin that's normal and one frataxin that's compromised, you're still fine, right? You need two compromised versions. But we all have some errors during, eventually everybody will have this disease, many, many years down the road, right? Because we're all making errors as we, not us, but our children's children. Right? Yeah. Well, we got to fix it now.
Let's fix it all.
Okay.
Yeah.
What did the separation process for these proteins look like? How did you obtain them from the raw material?
So we overexpressed them in E. coli. And actually, one thing I didn't tell you is we overexpress we take the genes for the NFS1 and the IST11, we put them on a plasmid, and we overexpress in E. coli. And actually, those two proteins grab the E. coli ACP and never let go, actually. Once that lipid inserts into the IST11, it's stuck there, right? And so then that material is how we purify it. And so that's also how the Sigla group purified it, right? The only difference between ours and their expression conditions was actually, one was just different types of media, essentially. And ours was actually a little cleaner, but it doesn't matter, you get crystals out of either form. So I don't know if that answered your question.
Final question for our speaker today? Final question for the semester?
Yeah.
You said that the frataxin disease usually occurs between age 5 and 15. How long does it take for that to become lethal? You said.
Depends on the mutant. And so there's two different forms. There's the form where you have the GAA repeat, and there's also a subset of the population that actually has mutations in the actual frataxin gene itself. And so we studied those as well. And depending on what mutants you have and how big your repeat is, determines how quickly the disease progresses. It's all about how much functional frataxin do you have. And so you just need enough to boost, so you can keep the iron sulfur assembly level at enough to satisfy your needs.
Well, if you have more questions, people will be here for a few minutes.